Hi all, welcome back. This is chapter 12. We're going to talk about how DNA is organized with the help of Alice, who usually abandons me as soon as I start broadcasting here. But we'll see how long she hangs around. She's a big fan of DNA. She's got her own. I would like you to go over your bio notes again about replication, your notes from genetics for replication. Consider what happens during the S phase of interphase from the beginning of our class, and then look over the structure of chromosomes. What is a centromere? What is the P arm? Those things before you start, just so we're all on the same page, because I want to talk to you about how it's all wrapped up and how we can add things or remove things to make either replication or transcription translation more accessible or less accessible. My plan is this is just one part today to look at the various structure of DNA and then distinguish between mechanisms that we use to make it easier or harder to copy. Look at how we actually look at it under an image. My bird is molting. Excuse me. Look at how we actually look at DNA banding in an image and then look at what those repetitive sequences are. I think we might be able to look at some of those repetitive sequences in lab, depending on what the research class is doing. I don't want to repeat exactly what they're doing. And then what is a pseudogene? So we look at the structure of chromosomes and we compare viral and bacterial chromosomes. We see that they have a fairly simple amount of DNA, but it's a lot. So both of these have been basically electrified so that their DNA falls out. This is a phage, so this is a T2 phage. Here it is. So they really do look like those little machines, and all of its DNA is fallen out. You can see that it has a massive amount of DNA that would be stored inside this little head here. Same thing for this E. coli. It's been zapped, sort of electrified, and all of its pores open, so its DNA falls out. It is a single molecule of DNA, a circle in both cases, and both of them are really devoid of a lot of the support proteins and structural support proteins, as well as the tags and ways we use as eukaryotes to turn genes on and off. They do it in different ways if they're going to access their genes. And for a virus, maybe all of the genes are on all of the time. In bacteria chromosomes, we do see their circular double-stranded nucleoid. So they have a piece of DNA that's a circle, and then of course because DNA is double-stranded, there's a complement to this strand, but that's all they have, and all their genes are on that one particular chromosome. They are wrapped up a little bit tight. They have some proteins that are associated with the DNA that are histone-like. I don't want to line here. Histones are the proteins that we use as eukaryotes to kind of wrap our DNA around to give it structural strength. They have something similar, but not exactly the same, and certainly not as many as we have. The DNA itself is super coiled. That means it's wrapped and then wrapped again. And if you imagine those sort of iPhone earbuds with the long white cords, when we had those, and everybody had them, if you just tossed them in your bag, 5 minutes after you took them off, they were completely and irrevocably tangled in a way that would take you forever to untangle them. The way to manage that is you took your earbuds and you kind of folded it and folded it and folded it and then wrapped the cord around tight, sort of like cord management for your earphones. You probably do this with rope in the real world as well. That super coiling or coiling and then coiling again helps to hold the structure of the molecule or the rope or the iPhone earbuds without actually risking breaking the molecule. Compact, contained, and hold on to the structure. Topoisomerases, not gyrase, but other topoisomerases, cut the DNA strands and then help with the winding and resealing. They repair any place that they break as those things wrap up really tight. Supercoiled DNA and topoisomerases are found in both eukaryotes and prokaryotes. It's kind of along the idea of if you want to keep a really long strand of something, you need to compact it and control it if you want to have something that is functional 5 minutes after you unwound it. You could rewind it. Think about yarn too. We don't keep yarn loose in a bag. you wind it up into a supercoiled structure. Polytene chromosomes are chromosomes that you can see with your own eyes. So this is part of the reason that fruit flies are such successful research organisms, is in their salivary glands and in other insect salivary glands as well. They have giant chromosomes. If you look at the chromosomes of a fruit fly, if we smush it and put their salivary glands under a microscope and stain it, you can literally see it with a light microscope. And that's what these are. And then they have these puff regions. This is a puff region on the top, which is areas where this DNA is coiled and supercoiled. But then the DNA itself that is actually being used in those regions is kind of left out in these little loop-de-loop puffs. Those loop-de-loop puffs are accessible to see things like RNA polymerases that are used during transcription. So making of these into mRNA so they can be used as protein instructions. They keep the unnecessary DNA for this particular cell wrapped up tight and puff out the necessary ones. And lamp brush, we're going to push it to the top here and talk about lamp brush chromosomes. These are large chromosomes with extensive looping. We see them in invertebrate egg making cells, so the oocytes, the cells that are going to become the eggs, and some insect spermatocytes. These areas, and you're actually seeing, look at these are chiasmata, but with genes that are looping out here. And these free loops are areas where the DNA is accessible. These are called lamp brush because I guess they look like brushes coming off. They're sites of activity where there's a lot of usage of these genes. The hard and fast rule to remember at this point is that if DNA is coiled up really, really tight, the enzymes that are used for transcription are not going to have access to that DNA. We can turn them off by coiling them tight or making them so that RNA polymerases cannot access them. If I want to wind up some DNA tight but have one gene that gets transcribed a lot, the way I can make that accessible to RNA polymerase is by kind of looping it out and leaving it flopping around. That way its promoter and all of the regions on that gene are accessible. Flip that. If I'm looking inside of a cell and I see these like brushes or I see areas where I have puffs, those are areas where the DNA is loose and hanging out. I can guess that those areas are areas of high transcription, making that into mRNA transcripts so I can make proteins from them. Chromatin, which we talked about at the end of the last video, chromatin is when the DNA is sort of loose and uncoiled in eukaryotes. It's decondensed, so it's not visible as a chromosome, and it's floating throughout the entire nucleus. This is what I'd see at interphase during G1, maybe even during S phase, before they condense into chromosomes to do the mitosis or the meiosis dance. They are wrapped around histones. Histones are proteins. These are not the same thing as histamines. A lot of students conflate them at this point. Histamines are part of your allergic response. They do sound similar. But histones are proteins that have a positive charge. Remember, DNA has a negative charge. They contain a lot of lysine and arginine. And we have 4 different kinds. I'm not asking you to memorize these. I'm asking you to look at the diversity. These are positive, so the DNA negative wraps tightly around them. And they have a very predictable structure. When I wrap my DNA around the histones here, this is a histone, and the red is the DNA here. I wrap them around histones and then bind them together in these little spherical particles like balls on a string or beads on a string. Those beads on a string here, and we looked at them at the end of lecture 11 as well. These little beads on the string are called nucleosomes, or inside the nucleus, nuclear bodies. Chromosomes are colorful bodies. Nucleosomes are nuclear bodies. These are all DNA wrapped around proteins. They're packed into solenoids, which are segments of these all wrapped together. So a solenoid is bigger than a nucleosome. A nucleosome is bigger than a histone. Histones are bigger than a single nucleotide, or the width of a piece of DNA. Your homework, I didn't put it on, I think I didn't put it on it, had an exercise where you organize these by size, which would have been super useful if it included, you were supposed to put it in the box by size. It didn't have anything labeled, so it was just kind of putting things in boxes, which didn't make any sense. I won't ask you the nanometer length of any of these, but I would like you to know what is bigger, 2 nanometers of the width of DNA, or a histone. It's a histone. A histone is smaller than a nucleosome. A nucleosome is smaller than looped DNA. Looped DNA is smaller than the one side piece of chromatin. Chromatin is smaller than chromatids. A chromosome is bigger than a chromatid. And this is a handy dandy guide to that. When chromosomes need to be repacked and resorted, we can move and access information by relaxing or adding access to my nucleosomes. The DNA is twisted and twisted into a superhelix with histones wrapped, and now these are the histones in this picture. In this case, there are multiple types of histones reflected by the different colors of the protein structures here. These histones kind of really look something like this as they're wrapped around each other. And so the DNA can wound around them like yarn wrapping around a cat's cradle, if you've ever done cat's cradle with yarn. I want you to notice here that the DNA is twisted and wrapped tight, but look at this little tail of the histone. Look at this tail sticking out, this tail sticking out. These little histone tails give me a mechanism for relaxing or tightening up the structure of this superhelix and this supercoil around the histone proteins. This little tail, I'm going to go with this one here, but there's one here, there's one here sticking out. These little tails that stick out, I can change the accessibility of this DNA to RNA polymerase by putting things on this tail. My options are an acetyl group. Adding an acetyl group or acetylation will open up this area. It makes it more non-polar. So acetyl groups are non-polar in their structure. Non-polar things like water, like non-polar molecules, but also non-polar things like RNA polymerase have more access to the molecule if it's sort of spreading and looping and free floating in the water. Adding acetyl to this tail, an acetyl group, makes the region that was wound around this tail more accessible to transcription. Methylation and phosphorylation serve similar functions. Methylation to a histone tail increases transcription. Phosphorylation, adding a negative phosphate group, actually kind of makes this DNA molecule a little more negative because I'm adding more negativeness to it. Negative things push each other away, and it opens up the molecule to be more accessible. Acetylation, methylation, and phosphorylation, the ation here just means the process of adding the first part of the word. Acetylation is the addition of an acetyl functional group. Boop. Methylation is the addition of a methyl group. Phosphorylation is the addition of a phosphate group. So fun fact, where we're going soon. If I methylate the DNA itself, not the histone tail, that decreases transcription. Methylation of the histone increases transcription as far as we understand right now. Methylation decreases it. We actually methylate an individual cytosines in the DNA. I'll talk about that as we get to that actual process, but those methylated cytosines then become inaccessible, possibly for the life of the organism, to transcription. Maybe not. 2 vocab words. I didn't really have a picture here because it would be the same picture again and it got old. I feel like it got old. Heterochromatin versus euchromatin. We have mentioned them. I want to define them officially. Heterochromatin, hetero means the same. So when it all looks the same under a scope, or when I stain it, or when I'm trying to understand it, when it all looks the same, it is very condensed. Condensed means squished together. When I see it during interphase, I can see it by staining it. It may, in the life of an organism or the life of a cell, some things may remain as heterochromatin all the time. The telomeres, so the ends on the end of each chromosome, tend to be wrapped really tightly all the time. They are heterochromatin. The centromere, that small little pinched in part in the center or kind of off to the side, depending on the chromosome. Here, this one looks like a planaria now. But these little pinched in bits are heterochromatin. Bar bodies and much of the Y chromosome during the lifetime of the organism are also just wrapped up really tight. They're not really used for transcription that much, if at all, and they are visible differently underneath staining. Euchromatin, U means true. Euchromatin is loose, it's flopping out. It's accessible by RNA polymerase, so it is transcribed, it's uncoiled, uncondensed, and active. And then when we stain it, it actually doesn't stain as well. So we can stain my nuclear material and see bands of hetero versus euchromatin by whether they take the stain or don't take the stain. It doesn't tell us what they do, but it tells us what is bound up tight versus what is loose and accessible. Like this. Here's some banding techniques. This is called C banding. It stains the centromere. There you go. This is centromere. This is heterochromatin. It is always wound up tight. You can see these guys are actually in the process of being copied. These little fellows, you can see their 2 sides here. It's just not a great picture with their little pinched in bit. C banding is centromere staining. G banding is actually what we've been looking at in the class all along as we've looked at chromosomes. It shows the complexity of heterochromatin versus euchromatin along the length. Heterochromatin stains very darkly, it stains. Euchromatin stains very lightly, it doesn't stain. So what I'm looking at here is not genes, but I'm looking at areas of lighter here, high transcription, all right, transcript, versus dark bands, which are going to be low or no transcription. And I want to, just in case, I've sang it a lot, but I haven't defined it. Transcription is the process of taking a DNA, and I think we talked about the central dogma, the taking DNA and forming mRNA. We're looking at the rate of transcription in these things, whether they're actually used to make mRNA, which is also then going to be translated and converted into a protein. If I look at chromosome banding on the X chromosome, so this is the X, it's fairly well understood. It allows us to identify different regions. This is the centromere, the mere little pinched in bit in the middle. It's heterochromatin. And then we can number the P arm, which is the short arm, and the Q arm, which is the long arm of my chromosome relative to the centromere and the ends. I am looking at banding regions here, so big banding regions. I'm not asking you to figure out how to label it. I'm going to just show you how you read it. Chromosome banding patterns are depending on chromosome banding patterns depend on the condensation, so how tightly wound each region of the chromosome is. This is not a bar body. This is an active X chromosome in this picture. Each chromosome band here contains up to about 10 million base pairs. So this is not a gene. This is a region of low transcription versus a region of high transcription. When I read this as P1 and 2 in this picture. This is the area above and below this really thick band. 1, this really thick band, and then 2. And then I read it by bands in here. So this is going to be the X chromosome, P, X, P, 1, 1, 4 refers to this band right here of higher transcription region. This band right here is XP 2, 1. XP 2, 1. Give me this one right here. What is this band called? And now you're looking up because you realize I stopped talking. What is this band called right here? That is XQ2, 4. So super easy once you see it. I have a little practice in the homework naming it. It's literally just the homework is missing this. When you do the homework, you can take this image with you and follow along so that you can just read the banding. There are a lot of repetitive DNA segments in eukaryotic DNA. Repetitive just means it's there as a fragment of DNA, A's and T's or C's and G's, a series of codes that are repeated over and over again. This has some evolutionary value. It certainly has value in identifying individuals. We're going to look at, I selected some. The textbook has all of these, and if we come and need them during lab, we'll come back and I'll repeat them as part of lab. I'll add them in. But I've just picked a couple here for you. Tandem repeats are one I know we're going to look at in lab, possibly satellite DNA. So to highlight again, centromeres are this big, tiny little pinched in piece of heterochromatin at the center of either a chromosome or a chromosome made out of sisters. The kinetochore proteins are when we are going to go through mitosis or meiosis. Little proteins comes off of the centromere. Because we don't want to pull on the centromere, it's actually made out of DNA and has potential coding inside of it. We want to pull on the molecule, it's the entire molecule itself. Let's replicate it so we do it right. And so that we're moving it by pulling it on proteins. The kinetochore proteins bind to the spindle fiber microtubules. The centromere are constrictions on the chromosome. Satellite DNA here are repetitive sequences of short repeats that makes up a depending on who you are, a variable portion of your total DNA. We find them in heterochromatin, so DNA that is not particularly used during the lifetime of an organism. They don't seem to be found in prokaryotes. I've got the fun side effect here is we're not really sure all the functionality or any of the functionality of this, but boy, there's a lot of them, so they might mean something. Here they are. I can tag them. I don't know what they mean necessarily, and that's going to be true as we move forward into our we're leaving classical genetics completely behind soon and moving into modern genetics. We can see these things, but we don't always know what they're good for. We can see variable number of tandem repeats. So this whole slide is about tandem repeats, but I have microsatellites, short tandem repeats versus variable number tandem repeats. These are repeating sequences of base pairs, so A, T, T, T. And then a repeat of A, T, T, T. We have a lot in eukaryotes of C, G, C, G, G, C, C, G, G. I'm sorry, in animals that are not insects. Insects have a lot of short tandem repeats of A's and T's. Again, we know these are important. They differ individuals from each other. If we can get the crime scene lab to work, and you can tell I'm not super optimistic about any of these things right now, but hey, if we get the crime scene things to work, what we're actually going to look at is the differences between individual criminals in the box is going to be the differences between these repeated segments that don't seem to be expressed, but they seem to matter in individuality. You might have different short tandem repeats than your parents. That tells me that you're a unique individual. We also have transposable elements. Transposable means I can move them from one place to another. They're mobile. That means they move around and can relocate within the genome. I have short interspersed and long interspersed transposable elements. The elements for ALU in humans seem to be the most obvious, noticeable. That's what they mean by prominent, short interspersed elements. These are elements, they don't code for ALU. They seem to respond to it, but again, I don't have an answer for why. The L1 elements are mostly prominent in the lines or the longer interspersed elements. These are dispersed through the genome. They're not tandemly repeated, but they do move. Retro, going backwards, transposons, moving. Trans means crossover, posons means they can move from place to place. There are some mobile transposable elements that are generated from RNA, and then they're encoded via reverse transcriptase, so transcription going backwards into DNA. These mobile transposons are called retrotransposons. Right now, because I don't have a great series of explanations of why these exist or what they are good for beyond the fact that we know they exist and they are interesting, I am only asking you to recognize the difference, and I'm going to go back here, between repetitive sequences. I want you to recognize the differences between satellite DNA, that these are variable, repeated, and found in heterochromatic regions. Tandem repeats, which are small repeated segments of DNA, whether they're really short or variable numbers, they're repeated sequences of the same code over and over again, versus transposable elements, these elements that are segments that seem to move from place to place. I do have a homework assignment on the homework, moving them into what kind of repeat they are. Use those slides to guide you through the answers to that. I think I have everything in the homework actually written on those slides. I am just trying to help you put them in 3 separate boxes at this point, and then maybe someday we'll be able to expand on them. Pseudogenes are the last thing I want to highlight here. These are single copy non-coding regions. They are significantly altered by insertions and deletions. They are representing vestigial evolutionary sequences, possibly showing connections to ancient ancestors, but because they're not coding, they're not really affected by natural selection. They don't do anything. They're just space inside the DNA. Maybe I can turn them back on and get ancestral ancient dinosaur DNA turned on inside of me, but it's likely that they've mutated away from anything that's functional at all. Our genome is surprisingly full of what looks like extra information. Maybe just 10% of our genome actually is coding for genes. a huge amount of it may be these pseudo means fake genes that are basically remnants of what we once were. And they definitely show in genetic analysis, if I'm building a phylogenetic tree or I'm trying to look at the evolution of creatures over time, they show connectiveness. But because they aren't controlled for by any natural selection mechanism, there's nothing stopping them from just mutating all to heck. And so we find a lot of mutations compared to what we would expect them to have been. They are not transcribed. They're just there. There's probably no pressure to get rid of them. So that's it. That's it for chapter 12. I've realized as I was filming this that I forgot to number this as 12. It was 11 when we started, but it's 12. It's definitely 12. As we understand more about genetics, I assume that this section will get longer. By the time you're teaching this, this will be a longer lecture. But I'm happy right now with you knowing that these things exist. The difference between acetylation, methylation, and phosphorylation right now is just which group we add, and that we're adding it to the histones in this case to increase transcription. The way we look at it is a little bit different, but we're looking at areas of high versus low or no transcription. And then at those repetitive segments, we don't have a lot. I don't have a lot to explain to you about repetitive segments because we know they exist and they may or may not be important. Or they may be like pseudogenes, where they are leftover fragments of our ancestry. And I will see you in class. I will see you in lab. We'll look at some P-GLOW this week, hopefully GLOW. And then we'll look at the structure of DNA on Thursday and head on from there. And I'll talk to you soon. All right. Bye-bye.